SIR Evolution Chapter 9. Chapter 9 Speciation. Divergent evolution describes the accumulation of distinct traits that can lead to speciation events. Speciation is the process by which new species form. A large population consists of a single ancestor species. Over time, different groups of the population come to inhabit different niches and develop traits for specialized inhabitants of that niche. As these changes accumulate, the population slowly develops distinct groups. When these groups can no longer reproduce due to some sexual barrier, a speciation event has occurred. This rapid increase in the number of species with a common ancestor, characterized by great ecological and morphological diversity, is termed as adaptive radiation. 9.1 Species concept. Species is a Latin word meaning kind or appearance. If one group of organisms consistently differs from other organisms, it will be defined as a separate species. Although the species is one of the most important units in biology, the definition of the term is still controversial. Most biological information is given with reference to a species. There is a whole discipline, taxonomy, dedicated to the delimitation of species, which requires a species concept. The taxonomist who describes the species will have examined specimens of it and of related species, looking for characters that are present in specimens of the species to be described and absent from other closely related species. Species concepts do not only define what a species is, but by defining what a species is, they also define what speciation is. New insights in the speciation process and the nature of species that accumulated in the past decade demand adjustments of the species concept. A first distinction among species concepts is between horizontal and vertical concepts. A horizontal concept aims to define which individuals belong to which species at any instant in time, e.g., biological, ecological, phentic species concept. A vertical concept aims to define which individuals belong to which species at all times, e.g. phylogenetic and evolutionary species concept. Biologists are mainly concerned with defining species in the present, and this requires a horizontal concept. We need to know which antelope are antelope cervicapra now, and are less interested in antelopes a million years in the past or the future. Here we will discuss some commonly used species concept to describe species. Table 25. 9.1.1. The biological species concept. The biological species concept is widely accepted. Species definition was first described by evolutionary biologist Ernst Mayr in 1942. He defined species as groups of populations that can actually or potentially exchange genes with one another and that are reproductively isolated from other such groups. The biological species concept defines species in terms of interbreeding. The biological species concept has been used to distinguish morphologically similar yet reproductively isolated species such as the northern leopard frog, Rana pipiens, and the southern leopard frog, R. utricularia. Despite its advantages, the biological species concept suffers from at least three limitations. 1. It has been noted that for many species with widely separate ranges, we have no idea if the reproductive isolation is by distance only or whether there is some species isolating mechanism. 2. Especially in plants, individuals called hybrids often form when parents from two different species are crossed with each other and the resultant progeny develop. This greatly blurs species distinctions. Oak trees provide a particularly good example of confusion in species definitions. Oaks from different species interbreed and their offspring are themselves viable, capable of reproducing with other oaks. For this reason, one might question whether the parental species should be called species at all. Ecologists have noted many examples of viable hybrid formation when historically isolated species are brought into contact through climate change, landscape transformation, or transport beyond their historic boundaries. 3. The biological species concept cannot be applied to asexually reproducing species such as bacteria and some plants and fungi, or to extinct species. 9.1.2 The recognition species concept. Patterson, 1993, defines a species as a set of organisms with a shared specific mate recognition system, SMRS. The specific mate recognition system is the sensory method by which organisms recognize potential mates. for example, as many as 30 or 40 different species of crickets may be breeding within a single habitat in India. The male crickets broadcast their songs and are approached by females. Interbreeding is confined within a species, because each species has its own distinctive song, and females only approach males that are singing their species song. The system of a male song and a female acoustic system that leads females to approach some songs and not others is an example of what is meant by an SMRS. The set of organisms that are defined as a species by the biological and recognition. Species concepts will be very similar, because organisms that interbreed will usually also have a shared SMRS. 9.1.3 Ecological Species Concept. American biologist Lee Van Valen, 1976, proposed the ecological species concept, in which each species occupies a distinct ecological niche, a unique set of habitat requirements. According to the ecological species concept, populations form the discrete phonetic clusters that we recognize as species because the ecological and evolutionary processes controlling how resources are divided up tend to produce those clusters. Competition between species is likely to result in each individual species occupying a unique niche. This species concept is useful in distinguishing asexually reproducing and morphologically similar species such as bacteria. 9.1.4 Phonetic Species Concept. The phonetic species concept is as an extension of the way taxonomists define species. Taxonomists define each species by a particular defining morphological character or characters that is shared by its members. In general, we could define a species as a set of organisms that are phonetically similar and distinct from other sets of organisms. This would be a phonetic species concept. It defines species in general by shared phonetic attributes. The classic version of the phonetic species concept is the typological species concept. The term morphological species concept has also been used to refer to much the same concept. The word typological comes from the word type, which is used in formal taxonomy. When a new species is named, its description is based on a specimen called the type specimen, which has to be deposited in a public collection. According to the typological species concept, a species consists of all individuals that look sufficiently similar to the type specimen of the species. A later version of the phonetic concept was developed by the School of Numerical Taxonomy in the 1960s. Numerical taxonomists developed statistical techniques for describing the phonetic similarity of organisms. Those techniques could be applied to recognize species. A species could then be defined as a set of organisms of sufficient phonetic distinctness. The limitation of phonetic species concept is that what must be the minimum phenotypic difference among two groups to be classified as separate species. The two biological species may differ by almost any phonetic amount. Another problem with this concept are sibling species, which are pairs of species that differ reproductively but not morphologically. Phonetic and reproductive units do not coincide in sibling species. 9.1.5 Phylogenetic species concept. Another popular definition of species is the phylogenetic species concept, which advocates that members of a single species are identified by a unique combination of characters. This definition incorporates the classic taxonomic view of species based on their morphological characters and, more recently, molecular features such as DNA sequences. A disadvantage of this concept is in determining how much difference between populations is enough to call them species. Using this definition, many currently recognized subspecies or distinct populations would be elevated to species status. 9.1.6 Evolutionary species concept. George Gaylord Simpson, 1961, proposed the evolutionary species concept whereby a species is distinct from other lineages if it has its own evolutionary tendencies and historical fate. For example, paleontologists have charted the course of species formation in the fossil record. One of the best examples documents the evolutionary changes that led to the development of many horse species, including modern horses. Some scientists believe this is both the best definition and, at the same time, the least operational, since lineages are difficult to examine and evaluate quantitatively. Incomplete fossil records and lack of transitional forms make lineages difficult to trace. Molecular scientists compare DNA sequences of particular organisms. However, it is difficult to decide how much of a difference in a DNA sequence separates groups into species. 9.2 Patterns or forms of speciation. The term evolution describes a change in genotypic frequencies, which usually results in a population of individuals better adapted to the environment than their ancestors were. Microevolution is the change in allele frequencies that occurs over time within a population. This change is mainly due to 4 different processes: mutation, selection, natural and artificial, gene flow, and genetic drift, which we have already discussed. Macroevolution involves studying patterns on the tree of life above the species level, e.g., new species, new genera, new families, and inferring the processes that are likely to have generated these patterns. Speciation, or the origin of new species, is a central process of macroevolution because any genus, family, or higher taxon originates with a new species novel enough to be the first member of the higher taxon. Speciation comes in 2 different forms as shown in Figure 84. 9.2.1 Anagenesis. Anagenesis, also known as gradualism, is a mechanism in which one species evolves into another without any splitting of the phylogenetic tree. Here species continue to exist and survive as an interbreeding population. It is also referred to as phyletic transformation and involves evolution within a single lineage where existing species gradually accumulates variation and with time becomes very different from parental species that now it can be regarded as new species. Lamarck was in support of anagenesis form of speciation. 9.22 Cladogenesis, branching evolution. Cladogenesis is a phenomenon of evolution that occurs by the divergence of taxa due to positive selection for the adaptation of sister populations of their common ancestor to different environments due to their anatomical, morphological, physiological, geographic, temporal, ecological, and/or ethological behavioral isolation. The budding of one or more new species from a parent species that continues to exist is more important than anagenesis in life's history because it is more common and can promote biological diversity. Darwin believes in this form of speciation. Figure 84. Forms of speciation. 9.2.3 Species formation may be gradual or sporadic. Concept of gradualism. The concept of gradualism proposes that new species evolve continuously over long periods of time. Figure 85a. It suggests large phenotypic differences that produce new species are the result of long periods of small genetic changes that accumulate over time. Such gradual transitions are relatively rare in the fossil record. Instead, fossils of new species appear relatively rapidly and with few transitional types. Concept of punctuated equilibrium. Stephen Jay Gould and Niles Eldredge, 1977, championed the idea of punctuated equilibrium, which suggests that the tempo of evolution is more sporadic. Figure 85b. According to this concept, species remain relatively unchanged for long periods of time and are at equilibrium with their environment. These long periods of stasis are punctuated by relatively rapid periods of change. This concept is supported by the fact that fossils of new species usually appear suddenly, with few transitional types. According to the idea of punctuated equilibrium, most evolution occurs not as ladders, in which one species slowly turns into another and turn into another, but as bushes, where species arise relatively quickly and where only a few active tips survive in the long term. Punctuated equilibrium predicts that a lot of evolutionary change takes place in short periods of time tied to speciation events. Punctuated equilibrium is an important but often misinterpreted model of how evolutionary change happens. Punctuated equilibrium does not suggest that Darwin's theory of evolution by natural selection is wrong. Does not mean that the central conclusion of evolutionary theory, that life is old and organisms share a common ancestor, no longer holds. Figure 85. The pace of speciation. Gradualism depicts evolution as a gradual change in phenotype from accumulated small genetic changes. The punctuated equilibrium depicts evolution as occurring during relatively long periods of stasis, punctuated by periods of relatively rapid evolutionary change. 5.3 Reproductive isolation. The mechanisms of reproductive isolation are a collection of evolutionary mechanisms, behaviors, and physiological processes critical for speciation. They prevent members of different species from producing offspring or ensure that any offspring are sterile. These barriers maintain the integrity of a species by reducing gene flow between related species. The mechanisms of reproductive isolation have been classified in a number of ways. Zoologist Ernst Mayr and Dobzhansky classified the mechanisms of reproductive isolation in two broad categories: prezygotic for those that act before fertilization or before mating in the case of animals, and postzygotic for those that act after it. The mechanisms are genetically controlled and can appear in species whose geographic distributions overlap, sympatric speciation, or are separate, allopatric speciation. 9.3.1 Prezygotic barriers. Prezygotic barriers prevents mating between species or impede formation of hybrid zygotes. 1. Habitat/ecological isolation. Two species that live in different habitats within the same area may encounter each other rarely, if at all even, though they are not technically geographically isolated. For example, two species of garter snakes in the genus Thamnophis occur in the same areas, but one lives mainly in water and the other is primarily terrestrial. Habitat isolation also affects parasites, which are generally confined to certain plant or animal host species. Two species of parasites living on different hosts will not have a chance to mate. We can assess sexual isolation in a species by bringing males and females from different populations and from the same population. Sexual isolation index will increase with geographic distance, i.e. males and female from different population, geographically isolated, will mate in very low frequency as compared to members of same population. The populations from different geographical region are genetically different. And we know as genetic distance increases, mating frequency decrease and sexual isolation index will increase. Figure 86. 2. Behavioral, ethological, sexual isolation. It represents special signals that attract mates, as well as elaborate behavior unique to a species. It is probably the most important reproductive barrier among closely related animals. The occurrence of different shape, coloration, habitat, and the range of overlapping regions may be some of the deciding factors in behavioral isolation. Male fireflies of various species signal to females of their kind by blinking their lights in particular patterns. The females respond only to signals characteristic of their own species, flashing back and attracting the males. The courtship rituals are specific to a species, and these courtship rituals are another form of behavioral isolation. The distinctive songs of many birds, the special mating calls of certain frogs, and the sexual displays of most animals are generally attractive only to mates of the same species. Many plants have floral displays that attract only certain insect pollinators. Even where the morphological differences between two species are minimal, behavioral differences may prevent cross-fertilization. Thus, Drosophila melanogaster and Drosophila simulans, designated as sibling species because of their morphological similarity, normally do not mate with each other, even when kept together in a single population cage. According to Coyne, male courtship in Drosophila group depends on their attraction to specific hydrocarbons in the female cuticle, and this behavioral isolation can be caused by only few genetic differences. 3. Temporal isolation. The time period of mating of different species are never always same. Two species that breed during different times of the day, different seasons, or different years cannot mix their gametes. This differential time period of mating, which prevents interbreeding between two different species, is known as temporal isolation. Temporal isolation in breeding seasons between closely related species leads to reproductive isolation. 3 species of the orchid genus Dendrobium living in the same rainforest does not hybridize because they flower on different days. Pollination of each species is limited to a single day because the flowers open in the morning and wither that evening. Similarly, different insect species may become reproductively active during different period. For example, Figure 87 below represents temporal isolation in breeding seasons among closely related sympatric species of insects. Species A is reproductive active during months of March to June, while species B is reproductive active during months of July to November. There is no overlap in reproductive activity, thus there will be reproductive barrier. 4. Mechanical isolation. Closely related species may attempt to mate, but cannot achieve the act or fail to achieve fertilization because their genitalia are anatomically not compatible or incompatible. This type of incompatibility, long thought to be a primary isolating mechanism in animals even, such kind of barriers contribute to reproductive isolation of flowering plants that are pollinated by insects or other animals. Floral anatomy is often adapted to certain pollinators that transfer pollen only among plants of the same species. Orchids of the genus Cryptostylis are known to maintain reproductive isolation because their flowers look and smell like females of the wasps of genus Lysipimpla. When the male wasp visits and attempts to mate with the flower, the shape of anther and stigma allows correct placement and transfer of pollen to the wasp, which then transfers the pollen to species-specific flower that it next attempts to mate with. This prezygotic barrier that prevents interspecies cross-pollination in Cryptostylis is best example of mechanical isolation through mimicry. 5. Gametic isolation. Even if the gametes of different species meet, they rarely fuse to form a zygote. For animals whose eggs are fertilized within the female reproductive tract, internal fertilization, the sperm of one species may not be able to survive in the environment of the female reproductive tract of another species. Many aquatic animals release their gametes into the surrounding water, where the eggs are fertilized. External fertilization. Even when two closely related species release their gametes at the same time in the same place, cross-specific fertilization is uncommon. Gametes recognition may be based on the presence of specific molecules on the coats around the egg, which adhere only to complementary molecules on sperm cells of the same species. In some Drosophila crosses have shown that an insemination reaction in the vagina of the female causes swelling and prevents successful fertilization of the egg. A similar mechanism of the molecular recognition prevents growth of pollen tube in the styles of foreign species. 9.3.2 Postzygotic barriers. If the prezygotic barriers do not impede the successful fertilization of the gametes belonging to different species, then postzygotic barriers prevent the hybrid zygote from developing into a viable, fertile adult. Postzygotic isolation usually follows Haldane's rule. J.B.S. Haldane, 1922, identified that when in the F1 offspring of the two different animal races one sex is absent, rare, or sterile, that sex is the heterozygous one. Thus, hybrids of the heterogametic sex have lower fitness than hybrids of the homogametic sex. Haldane's rule only says that when fitness differs between the sexes of hybrid offspring, the heterogametic sex has lower fitness. According to Dobzhansky-Muller theory, postzygotic isolation is caused by interactions between genes at many loci. According to it, speciation happens as an almost automatic consequence of ordinary selection and drift within a population, as each population evolves in its own environmental conditions. Speciation does not require special conditions, in which normal evolutionary processes are suspended. The important post-zygotic isolation mechanisms are 1. Reduced hybrid viability or hybrid inviability. When prezygotic barriers are crossed and hybrid zygotes are formed, genetic incompatibility between the two species may abort development of the hybrid at some embryonic stage. Of the numerous species of frog belonging to the genus Rana, some live in the same regions and habitats, where they may occasionally hybridize, but the hybrids generally do not complete their development. 2. Reduced hybrid fertility or hybrid sterility. Even if two species mate and produce the hybrid offspring that are vigorous, reproductive isolation is intact if the hybrids are completely or largely sterile. Since the infertile hybrids cannot backbreed with either parental species, genes cannot flow freely between the species. 1. Cause of this barrier is a failure of the meiosis to produce normal gametes in the hybrid if chromosomes of the two parent species differ in number or structure. A familiar case of a sterile hybrid is the mule, a robust cross between horse and a donkey. Horses and donkeys remain distinct species, because, except very rarely, mules cannot backbreed with either species. 3. Hybrid breakdown. In some cases when species crossmate, the first-generation hybrids are viable and fertile, but when these hybrids mate with one another, or with either parent species, offsprings of the next generation are feeble or sterile. For example, different cotton species can produce fertile hybrids, F generation, but breakdown occurs in the next generation, F2 generation, when offspring of the hybrids die as seeds or grow into weak and defective plants. 9.4 Mechanisms of speciation. There are 4 general mechanisms of speciation based on different geographic conditions and how the gene flow is interrupted in the speciating populations. Figure 88. If a new species evolves in geographic isolation from its ancestors, the process is called allopatric speciation. If the new species evolves in a geographically contiguous population, it is called parapatric speciation. If the new species evolves within the geographic range of its ancestor, it is called as sympatric speciation. If small founder populations colonize an area outside the main population range and forms new species, it is termed as peripatric speciation. 9.4.1 Allopatric speciation. The appearance of a geographic barrier, such as a river or mountain, through the range of a species, physically isolates populations of the species. The physically isolated populations can then evolve independently. If reproductive isolating mechanisms evolve, then 2 distinct species are formed, and if they come together in the future, they remain distinct species. Speciation that occurs because reproductive isolating mechanisms evolve during physical separation of the populations is called allopatric speciation. Reproductive isolating mechanisms usually originate incidentally to the speciation process. That is, they arise incidentally during the process of evolution in isolated populations rather than being selected for. When isolated populations come together again, incomplete isolating mechanisms may allow hybrids to form. If the hybrids are normal and viable and can freely interbreed with individuals of each parent population, then no speciation has taken place. However, if the hybrids are at a disadvantage, natural selection may favor stronger isolating mechanisms. In this case, organisms that mate with individuals from the other population leave fewer offspring. The result is a more effective barrier to hybridization. Regions in which previously isolated populations come into contact and produce hybrids are called hybrid zones. 9.4.2 Parapatric speciation. Parapatric speciation occurs at adjacent places due to variations in environmental gradients. Although no physical barrier arises, the new niche acts as a barrier to gene flow between the population in the new niche and the rest of the species. Parapatric speciation is believed to have occurred often in relatively non-vagal animals such as snails, flightless grasshoppers, and annual plants. In parapatric speciation, the new species evolve from contiguous populations rather than completely separate ones, as in allopatric speciation. The full process could occur as follows. Initially, one species is distributed in space. The species evolves a stepped cline pattern of geographic variation. The stepped cline could exist because of an abrupt environmental change. One form of the species would be adapted to the conditions on one side of the boundary, the other form to the conditions on the other side of the boundary. A hybrid zone is a stepped cline in which the forms on either side of the boundary are sufficiently different that they can easily be recognized. The conditions in a hybrid zone or a stepped cline are particularly favorable for speciation if it is a tension zone. A tension zone exists when the hybrids between the forms on either side of the boundary are selectively disadvantageous. The two forms may have been given different taxonomic names as subspecies or races, or they may be different enough to have been classified as separate species. An example of parapatric speciation can be discussed in a scenario given as follows. A species of grass grows around a mine area having patches of heavy metal-contaminated soil. Some of the populations of the species grew selectively on the soil that was contaminated with heavy metals. Over a period of time, though the tolerant and non-tolerant grass populations were continuously distributed and not separated by geographical barriers, they eventually evolved different flowering times and became different species. This process of speciation would be termed as parapatric speciation. 9.4.3 Sympatric speciation. In sympatric speciation, speciation occurs in populations that live in the same geographic area, although such contact makes sympatric speciation less common than allopatric speciation. Sympatric speciation can occur if gene flow is reduced by such factors as polyploidy, habitat differentiation, pathogen and sexual selection. Reproductive isolation can be introduced in host due to infection of pathogens. For example, Wolbachia are obligate intracellular bacteria, many different strains of which are abundantly present in insects. They induce mating incompatibility in insect host, i.e. males infected with one strain can only fertilize females infected with the same strain. No other pathological effects are observed in host. Under such scenario, reproductive isolation will be introduced among insects, which can lead to rapid sympatric speciation. A species may originate from an accident during cell division that results in extra sets of chromosomes, a condition called polyploidy. There are two distinct forms of polyploidy. An autopolyploid is an individual that has more than two chromosome sets that are all derived from a single species. A second form of polyploidy can occur when two different species interbreed and produce hybrid offspring. Various mechanisms can change a sterile hybrid into a fertile polyploid called an allopolyploid. Sympatric speciation can also occur when genetic factors enable a subpopulation to exploit a habitat or resource not used by the parent population. Phytophagous insects may also split sympatrically by host shifts. 9.4.4 Peripatric speciation. Peripatric speciation is a form of speciation, the formation of new species through evolution. In this form, new species are formed in isolated peripheral populations. This is similar to allopatric speciation in that populations are isolated and prevented from exchanging genes. However, peripatric speciation, unlike allopatric speciation, proposes that one of the populations is much smaller than the other. 5. Adaptive radiations. The rate at which loss and gain of new life forms occurs affects the diversity of life. A species is lost due to extinction, a species is gained, or new species is formed by speciation events, whereas the loss of species is by extinction. Diversity of a population increases when the extinction rate exceeds the speciation rate. When a novel characteristic is evolved, it creates a new adaptive zone, which is a set of new living conditions and resources that presents many previously unexploited opportunities. This gives a dimension for new adaptive radiations. For example, many new evolutionary possibilities emerged with the evolution of wings of insects, such as rapid access to treetops, islands, and other new feeding and breeding areas. Adaptive radiation can occur at all taxonomic levels and on all geographic scales. For example, the proliferation of life on Earth from the origin of life to the present is an adaptive radiation on the largest scale. However, adaptive radiations are particularly clear when they occur in a relatively small taxonomic group, in a confined geographic area. For example, different species of finches on the Galapagos Islands are evolved with a range of ecological adaptation from Darwin's finches. Adaptive radiations occur in a number of circumstances. 1. Colonization of a new area where there are no competitors. As an example, when the first ancestral species colonized the respective area on the Galapagos Islands, the radiation of Darwin's finches on island chains occurred. Probably no competitors were present, because the island had only recently emerged from the sea. The new area contains unexploited resources, and the ancestral species radiated into a range of forms that could exploit these resources. 2. Extinction of competitors. This can be best explained with the example of colonization and radiation of mammals just after the extinction of dinosaurs. Mammals could occupy the space vacated by the extinction of dinosaurs. 3. Replacement of competitors. If organisms of a taxon are adaptively superior to the members of other taxon, generally they radiate more than the other species. Superior will take over from the inferior one, which will be driven extinct. The superior taxon may have become superior because of environmental changes or because it evolved a new, superior adaptation. 9.4.3 Sympatric speciation. In sympatric speciation, speciation occurs in populations that live in the same geographic area, although such contact makes sympatric speciation less common than allopatric speciation. Sympatric speciation can occur if gene flow is reduced by such factors as polyploidy, habitat differentiation, pathogen, and sexual selection. Reproductive isolation can be introduced in host due to infection of pathogens. For example, Wolbachia are obligate intracellular bacteria, many different strains of which are abundantly present in insects. They induce mating. Incompatibility in insect host, i.e. males infected with one strain can only fertilize females infected with the same strain. No other pathological effects are observed in host. Under such scenario, reproductive isolation will be introduced among insects, which can lead to rapid sympatric speciation. A species may originate from an accident during cell division that results in extra sets of chromosomes, a condition called polyploidy. There are two distinct forms of polyploidy. An autopolyploid is an individual that has more than two chromosome sets that are all derived from a single species. A second form of polyploidy can occur when two different species interbreed and produce hybrid offspring. Various mechanisms can change a sterile hybrid into a fertile polyploid called an allopolyploid. Sympatric speciation can also occur when genetic factors enable a subpopulation to exploit a habitat or resource not used by the parent population. Phytophagous insects may also split sympatrically by host shifts. 9.4.4 Peripatric speciation. Peripatric speciation is a form of speciation, the formation of new species through evolution. In this form, new species are formed in isolated peripheral populations. This is similar to allopatric speciation in that populations are isolated and prevented from exchanging genes. However, peripatric speciation, unlike allopatric speciation, proposes that one of the populations is much smaller than the other. 5. Adaptive radiations. The rate at which loss and gain of new life forms occurs affects the diversity of life. A species is lost due to extinction, a species is gained, or new species is formed by speciation events, whereas the loss of species is by extinction. Diversity of a population increases when the extinction rate exceeds the speciation rate. When a novel characteristic is evolved, it creates a new adaptive zone, which is a set of new living conditions and resources that presents many previously unexploited opportunities. This gives a dimension for new adaptive radiations. For example, many new evolutionary possibilities emerged with the evolution of wings of insects, such as rapid access to treetops, islands, and other new feeding and breeding areas. Adaptive radiation can occur at all taxonomic levels and on all geographic scales. For example, the proliferation of life on Earth from the origin of life to the present is an adaptive radiation on the largest scale. However, adaptive radiations are particularly clear when they occur in a relatively small taxonomic group, in a confined geographic area. For example, different species of finches on the Galapagos Islands are evolved with a range of ecological adaptation from Darwin's finches. Adaptive radiations occur in a number of circumstances. 1. Colonization of a new area where there are no competitors. As an example, when the first ancestral species colonized the respective area on the Galapagos Islands, the radiation of Darwin's finches on island chains occurred. Probably no competitors were present, because the island had only recently emerged from the sea. The new area contains unexploited resources, and the ancestral species radiated into a range of forms that could exploit these resources. 2. Extinction of competitors. This can be best explained with the example of colonization and radiation of mammals just after the extinction of dinosaurs. Mammals could occupy the space vacated by the extinction of dinosaurs. 3. Replacement of competitors. If organisms of a taxon are adaptively superior to the members of other taxon, generally they radiate more than the other species. Superior will take over from the inferior one, which will be driven extinct. The superior taxon may have become superior because of environmental changes or because it evolved a new, superior adaptation. 4. Adaptive breakthroughs. New adaptations in a taxon may enable them to compete with another taxon, or may allow it to exploit a previously unexploited resource. 4. Instance animals which evolved appropriate adaptations colonized the land as. The colonization of land took place in a similar manner as the new island chain, point 1 above. However, we can distinguish between colonization of an existing area, made by the evolution of a new adaptation, and colonization of a new area that requires no new adaptation. The basic requirements for an organism to colonize on land are evolution of adaptation for support, respiration, and water retention. By contrast, the finch that first colonizes the Galapagos Island probably did not have to evolve a new adaptation before it could colonize the island. Adaptive breakthroughs generated new extensive adaptations after colonization of land. 6. Divergent and convergent evolution. The American naturalist J. T. Gulick, 1832–1923, was the first to use the term divergent evolution. Divergent evolution is the accumulation of differences between closely related populations within a species, leading to speciation. Divergent evolution is typically exhibited when two populations become separated by a geographic barrier, such as an allopatric or peripatric speciation, and experience different selective pressures that drive adaptations to their new environment. After many generations and continual evolution, the populations become less able to interbreed with one another. Classic examples of divergence in nature are the adaptive radiation of the finches of the live in different Galapagos Island. The different species formed as a result of divergent evolution since share common ancestry have various homologous organs and homologous genes. Both orthologous genes, resulting from a speciation event, and paralogous genes, resulting from gene duplication, can illustrate divergent evolution. Through gene duplication, it is possible for divergent evolution to occur between two genes within a species. Similarities between species that have diverged are due to their common origin, so such similarities are homologies. Convergent evolution is the independent evolution of similar features in species of different periods or epochs in time. As a result of this, unrelated or distantly related organisms evolve similar body forms, coloration, organs, and adaptations. Convergent evolution creates analogous structures that have similar form or function but were not present in the last common ancestor of those groups. The cladistic term for the same phenomenon is homoplasy. Natural selection can result in evolutionary convergence under several different circumstances. Organisms displaying these similarities usually live in similar environments, and the force driving convergence is natural selection. Similar environments pose similar challenges to survival, and traits that aid in survival are selected for in each environment. Species can converge in sympatry, as in mimicry complexes among insects, especially butterflies. Other examples include coral snakes and their mimics. Mimicry evolves after one species. The model has become aposematic, warningly colored, because it is toxic or poisonous, and therefore protected. Two distinct kinds of mimicry are recognized: Batesian and Müllerian. In Batesian mimicry, the mimic is palatable or unprotected, but gains from being mistaken for the model, which is unpalatable or protected. Two protected model species can also converge because of the advantage of being mistaken for each other. Müllerian mimicry. Evolutionary convergence involving unrelated organisms living in similar environments but in different places, allopatry, can also occur in another way. This usually takes place in relatively simple communities in which biotic interactions are highly predictable and the resulting number of different ways of exploiting the environment are limited. Similar environments pose similar challenges to survival and reproduction, and those traits that enhance Darwinian fitness are selected for in each environment. Such organisms that fill similar ecological roles in different, independently evolved biotas are termed ecological equivalent. Examples of convergent evolution. Wings and wing-like structures have evolved independently several times in insects, reptiles, pterosaurs, and birds, and in mammals, bats. Flight first evolved in insects about 330 million years ago, Maya, second in pterosaurs, about 225 Maya. Later in birds, about 150 Maya, and still later in bats, 50 to 60 Maya. Some frogs, lizards, and mammals have also evolved the ability to glide, presumably a precursor to flight. In order to land safely, such hang gliders must time their stall precisely at the right moment and place. Convergent evolution is seen in the fusiform, tapering toward the end, shapes and similar counter-shading coloration of sharks and dolphins, both of which are adapted to marine environments. Their shape facilitates rapid and efficient movement through water, and their light underbelly and a gray upper surface make them less visible from both below and above. Convergent evolutionary responses of lizards to arid environments are evident between continents, for example, Australian and North American deserts both support a cryptically colored and thornily armored ant-specialized species. The thorny devil, Moloch horridus, an agamid, exploits this ecological role in Australia, while its counterpart, the desert horned lizard, Phrynosoma, an iguanid, occupies it in North America. No Kalahari lizard has adopted such a lifestyle. Interestingly, morphometric analysis demonstrates that the thorny devil and desert horned lizard are actually anatomically closer to one another than either species is to another member of its own lizard fauna, which are much more closely related. The pair of the species with morphogoical and niche similarity, without any common ancestry, demonstrate the concept of convergent evolution between the South America and Africa. Arid regions of South Africa support a wide variety of euphorbiaceous plants, some of which are strikingly close to American cacti. Phenotypically, they are leafless stem succulents, protected by sharp spines, presumably adaptations to reduce water loss and predation in arid environments. A very striking example of evolutionary convergence involves the eyes of vertebrates and cephalopod mollusks. Both have independently evolved complex camera-like eyes complete with an aperture, lens, and retina. Although vertebrate eyes are poorly designed as compared to cephalopod eyes. In vertebrates, nerve fibers pass in front of the retina, creating a blind spot, whereas nerves lie behind the retina in the superior cephalopod eye, which does not have a blind spot. 9.7 Coevolution. In its broadest sense, coevolution refers to the joint evolution of two or more taxa that have close ecological relationships but do not exchange genes, and in which reciprocal selective pressures operate to make the evolution of either taxon partially dependent on the evolution of the other. Thus, coevolution includes most of the various forms of population interaction, from competition to predation to mutualism. Coadaptations, such as those between an ant, Formica fusca, and a caterpillar of the lichenid butterfly, Glaucopsyche ligdamus, likely arise by coevolution between the lineages leading to the two modern species. The caterpillars are parasitized by braconid wasps and tachinid flies. Alone, they are almost defenseless against these lethal parasites, but the mutualistic ants will fight off parasites from their caterpillars. In return, caterpillar provides honeydew from a special organ, newcomer's organ, as a food for ants. Natural selection probably favored mutually adapted changes in each species after the ancestors of the two species had become associated. Changes in one species, such as to increase honeydew production, would favor changes in the other to increase protection, as the caterpillars became more beneficial to the ants. This kind of reciprocal influence is what is meant by coevolution. Each species exerts selection pressures on and evolves in response to the other species. The two lineages evolve together. Figure 89. Figure 89. Coevolution means that two separate lineages mutually influence each other's evolution, and they evolve together. The Red Queen hypothesis is related to the coevolution of species. It was coined by Lee Van Valen to explain that a species must adapt and evolve not just for reproductive advantage, but also for survival because competing organisms also are evolving. In other words, it states that species must constantly adapt and evolve to pass on genes to the next generation, and also to keep from going extinct when other species within a symbiotic relationship are evolving. We look at several examples of coevolution. In all examples, a species evolves in relation to each other. One interesting example of coevolution is between fruit-eating birds and fruit-bearing plants. Many fruit-eating birds, especially in tropical rainforests, are co-evolving with the plants whose fruits they eat. The birds get nourishment, and in the process the plants get their digestion-resistant seeds dispersed by regurgitation or along with the birds' droppings. Many characteristics of the plants have evolved to facilitate dispersal, and the behavior and diets of the birds have responded to those changes. In particular, the plants have evolved conspicuously colored, relatively odorless fleshy fruits to attract the avian dispersers of their seeds. They are co-evolving in response to the finely honed visual systems of the birds. Plant species co-evolving with colorblind mammalian seed dispersers have, in contrast, dull-colored but smelly fruits. The bird-dispersed plants often have evolved fruits with giant seeds covered by a thin, highly nutritious layer of flesh. This forces the bird to swallow the fruit whole, since it is difficult or impossible just to nip off the flesh. In response, birds that are specialized frugivores, that is, that do not take other kinds of food, have evolved both bills with wide gapes, so they can swallow the fruit whole, and digestive tracts that can rapidly dissolve the flesh from the large impervious seed, which then can be regurgitated. Coevolution are always mutualistic, even it can be antagonistic. Antagonistic coevolution can have various forms such as prey-predator relationship, host-parasite interaction, or plant secondary metabolite and associated insects. A plant may evolve a secondary chemical substance that deters the vast majority of predators, but if a particular herbivore can in turn evolve a physiological means of coping with the chemical deterrent, it can thereby obtain an uncontested food supply. Through this kind of coevolution, many herbivores have become strongly specialized on a single species or a few closely related species of plants. Thus, Drosophilopatria is the only species of fruit fly that can exploit the Cnidae group of cacti. These plants produce an alkaloid that is fatal to the larvae of all other fruit flies, but Dipatria has evolved a means of detoxifying this chemical. In many cases, such specialized herbivores even use a plant's toxic chemicals, often aromatic and quite pungent, as cues in locating and/or selecting their host plants. Some herbivores, such as the monarch butterfly, actually sequester plant poisons, cardiac glycosides in this case, which in turn make the herbivore itself unpalatable or even poisonous to its own potential predators. Coevolutionary arms races can result in evolutionary escalation. Suppose that certain individuals in a population of carnivores, such as hawk, become better at hunting prey, such as mice. Because of genetic variation, certain individuals of prey have traits that allow them to escape or hide from their predators, and they pass these adaptive traits on to some of their offspring. However, a few individuals in the predator population also may have traits, such as better eyesight or quicker reflexes, that allow them to hunt the better adapted prey successfully. They would then pass these traits on to some of their offsprings. Step-by-step coevolution is particularly likely to take place between parasites and their hosts. They can have specific and close relations, and it is easy to imagine how a change in a parasite, which improves its ability to penetrate its host, will reciprocally set up selection for a change in the host. 9.8. Sexual selection. Sexual selection was Darwin's explanation for the evolution of apparently useless but conspicuous traits in males of many species, such as bright colors, long tails, horns, antlers, and elaborate courtship displays. He hypothesized that these traits either improved the ability of their bearers to compete for access to members of the other sex, intersexual selection, or made their bearers more attractive to members of the other sex, intersexual selection. Darwin argued that female preferences for such features are also the result of sexual selection because, unornamented or unattractive, males would succeed equally in the battle for life and in leaving a numerous progeny, but for the presence of better endowed males. Sexual selection may result in species that are sexually dimorphic, that is, species in which males and females differ in size, shape, or color. The concept of sexual selection was not well received by Darwin's contemporaries. However, Many examples of sexual selection have been investigated in the century and a half since he first proposed the idea, and Darwin turned out to be right. For example, sexual selection is responsible for different morphological attributes of male birds that compete with each other for available females. One case in point is the remarkable tails of male African long-tailed widowbirds, which are longer than their heads and bodies combined. To examine the role of sexual selection in the evolution of widowbird tails, a behavioral ecologist captured some male widowbirds. He shortened the tails of some males by cutting them and lengthened the tails of others by gluing on additional feathers. Male widowbirds normally select and defend from other males, a site where they perform courtship displays to attract females. Both short-tailed and long-tailed males successfully defended their display sites, indicating that a long tail does not confer an advantage in male-male competition. However, males with artificially elongated tails attracted about 4 times more females than did males with shortened tails. Figure 90. 9.8.1 Long tail is an honest signal of good relative fitness. The ability to grow and maintain a costly feature, such as a long tail, may indicate that the male bearing it is vigorous and healthy. Hypothesis that having well-developed ornamental traits signals vigor and health has been tested experimentally with captive zebra finches. The bright red bills of male zebra finches are the result of red and yellow carotenoid pigments. Zebra finches and most other animals cannot synthesize carotenoids and must obtain them from their food. In addition to influencing bill color, carotenoids are antioxidants and components of the immune system. Males in good health may need to allocate fewer carotenoids to immune function than males in poor health. If so, then females can use the brightness of his bill to assess the health of a male. Investigators manipulated blood levels of carotenoids in male zebra finches by means of carotenoid supplements. Experimental males were given drinking water with carotenoids added. Control males were given only distilled water. All the males had access to the same food. After 1 month, the experimental males had higher levels of carotenoids in their blood, had much brighter bills than the control males, and were preferred by female zebra finches. Next, the investigators challenged both groups of males immunologically by injecting phytohemagglutinin, PHA, into the webs of their wings. PHA induces a response by T lymphocytes, resulting in an accumulation of white blood cells and thus a thickening of the skin. Experimental males with enhanced carotenoid levels developed thicker skins because they responded more strongly to PHA than control. Males did, indicating a heightened immune system. This experiment showed that when a female chooses the male with a bright red bill, she probably gets a mate with a healthy immune system. Such males are less likely to become infected with parasites and diseases, so they are less likely to pass on infections to their mates. Healthier males are also better able to assist with parental care than are males with duller bills. Experiment. Hypothesis. Sexual selection favors the evolution of long tails in African long-tailed widowbirds. Method. Capture males and artificially lengthen or shorten their tails by cutting feathers or gluing on feathers. Figure 90. The role of sexual selection in the evolution of widowbird tails. 9.8.2 Zahavi Handicap Principle. It is a hypothesis proposed by Amat Zahavi to explain how evolution may lead to honest or reliable signaling between animals which have an obvious motivation to bluff or deceive each other. It suggests that costly signals must be reliable signals, costing the signaler something that could not be afforded by an individual with less of a particular trait. For example, in sexual selection, the theory suggests that animals of greater biological fitness signal this status through handicapping behavior or morphology that effectively lowers this quality. The central idea is that sexually selected traits function like conspicuous consumption, signaling the ability to afford to squander a resource. Receivers then know that the signal indicates quality, because inferior quality signalers are unable to produce such wastefully extravagant signals. 9.8.3 Assessing the costs of adaptations. As we mentioned at the beginning of this chapter, adaptations typically impose costs as well as benefits, and the evolution of adaptations depends on the trade-off between those costs and benefits. Garter snakes in some populations, for example, can eat rough skin newts without being poisoned, but they pay for this ability by sacrificing crawling speed. Determining the costs and benefits of a particular adaptation is difficult because individuals differ not only in the degree to which they possess the adaptation but also in many other ways. The cost of long tails was not measured in the experiments with widowbirds, but related studies have been done on males of other species. In some mammalian species, including deer, lions, and baboons, one male controls reproductive access to many females. These polygynous species tend to be sexually dimorphic. The males appear quite different from the females. Males of these species are significantly larger than females and often bear large weapons, such as horns, antlers, and large canine teeth. Size and weaponry are needed to defend a male's multiple mates against other males of the species. The costs of sexual dimorphism for males of polygynous species were assessed using the comparative method. Such males have higher parasite loads and higher mortality rates than females of their own species because maintaining a large size and bearing large weapons makes them more susceptible to parasites. In addition, when compared to parasite loads in males of closely related monogamous species, in which males and females are essentially monomorphic, appearing quite similar, the dimorphic males carried higher parasite loads in almost every case. 9.8.4 Fisherian runaway or runaway selection. It is a sexual selection mechanism proposed by the Ronald Fisher to account for the evolution of exaggerated male ornamentation by persistent, directional female choice. An example is the colorful and elaborate peacock plumage compared to the relatively very short and dull-colored peahen plumage. Fisher developed the theory further by assuming genetic correlation between the preference and the ornament. That initially the ornament signaled greater potential fitness, the likelihood of leaving more descendants, so preference for the ornament had a selective advantage. Subsequently, if strong enough, female preference for exaggerated ornamentation in mate selection could be enough to undermine natural selection even when the ornament has become non-adaptive. Over subsequent generations, this could lead to runaway selection by positive feedback, and the speed with which the trait and the preference increase could, until counter-selection interferes, increase exponentially.